Bismillahirrahmanirrahim. Welcome back, guys. Welcome to another video sessions from Kame Biology. In this video lecture, we will talk about the life cycle of the adenoviruses. Now, adenoviruses are those viruses which can cause cancer inside the human body. Fine. Adenoviruses are first of all isolated from the adenoid tissue, so that's why the name are known as adenoviruses. Fine. Adenoviruses are actually they are non-enveloped viruses, means they are naked viruses, or adenoviruses are look like in icosahedral forms, or we can say that they are icosahedral viruses. How the adenoviruses start their life cycle and how they can infect the host cell, and inside the host cell how they can start their life cycle. So we will all talk about this replication schemes of the adenoviruses in this video. So stay here and continue to watch. Now let me write the life cycle of the adenoviruses. So in this video we will talk about the life cycle of the adenoviruses. Life cycle of adenovirus. Adenoviruses, they usually cause what? They cause cancer, or we can say they can show the properties of oncogenomic viruses. Cancer, they can cause what? They cause cancer inside the human body. Fine. So adenoviruses, they don't have envelope. So now let me draw the structure of the adenoviruses. So how it will be look like? So it will be look like in this form. Means the adenoviruses are actually in icosahedral form. Fine. Instead of this envelope area, they have some spikes proteins. So now let me draw. These are the receptor proteins. Fine. And these receptor proteins are responsible for the attachment or for the interaction to the host cell, host membrane. Fine. Host cell membrane. Now this hydrophobic site will called as a fiber, and this one side is called as a pentone. This one area, this capsid area contains some other proteins. Means this capsid area will contain some proteins as I draw in a blue form. So these are usually called what? Hexone. Fine. And the adenovirus are actually, they are the double-stranded DNA viruses. They are the double-stranded DNA viruses. These adenoviruses are isolated from the adenoid tissue. Where the adenoid tissue are present, the adenoid tissue are present inside the mode, there are the alva. Now on the upper side of the alva, adenoid tissues are present. From the adenoid tissue, these viruses are isolated. Fine. So what happened? First of all, the adenoviruses are present in the infected person, means viral infected person, and they will carry the adenoviruses. So once the healthy person will present from this infected person almost 5 foot, so they will easily get the adenoviruses. So once the healthy person will get this adenoviruses, so the adenoviruses will first enter into the mouth, and then they will reach to the alva, and from the alva they will reach to the adenoid tissue, and from the adenoid tissue they will attach themselves to the host cell, means adenoid cells, and then they will cause the replication schemes. Fine. So now let me draw the host cell here. Let's suppose these are the host cell, and with the host cell there are also some receptors are arises, and we call that core family receptors. Core family receptor. Fine. These are the core family receptors. Now what happened? The adenoviruses means the receptors of the adenovirus, means the spikes of the adenoviruses will attach to this receptors of the adenoid, or with the host cell receptors. Once they will attach with the host cell receptor, so what happened? they will be internalized. So the whole viruses will be engulfed by this host cell. So only the protein area, means this spikes area, will remain outside, and just the virus, means this capsid area, will be internalized. So only virus will be internalized, and which carry the double-stranded DNA. And after that, what happen? They will then reach to the nucleus, or first they will migrate inside the microtubules, moves inside microtubules. Now once they will move inside the microtubules, and from the microtubules they will reach to the host nucleus. So now let me draw the host nucleus with a blue marker. So these are the host nucleus, and these are the pore. So they will enter inside the pore, and this capsid area will remain outside. So the capsid area will remain outside, and just the genomic, means the DNA, will be internalized. One thing more that I want to tell you, I forget to mention that this host means this viral genome contains some proteins, terminal proteins, and we call that 55 kd terminal protein. It contains 55 kd terminal proteins. Now what happened after that? There are also the polymerase enzyme are also present inside this nucleus. These polymerase enzymes, RNA polymerase enzymes, are responsible to produce messenger RNA. But before that, first of all, what happened? So from this DNA, they will produce the messenger RNA. What they will produce? The early mRNA. Now this early mRNA are then translated, means translation process will occur, and then we get what? We get proteins. So we get a lot of proteins. And among these proteins, these are actually E1, E3 and E4 proteins. Now the function of the E1 proteins to induce a transcription. They will induce a transcription. E1 protein means the E1 enzyme. The function of E3 and E4 are used in our DNA replication, because we need the DNA replication in the adenoviruses. Fine. So E1 protein are responsible in our DNA replication processes. Now what happened? After that, once these proteins are generated, then what happened? This, if I draw the strain in a linear form, these are the terminal proteins, mean 55 kd proteins. Fine. First of all, what happened? This upper strand is used as a template, and once this upper strand is used as a template, then from this upper strand we get the new copy of the DNA. So we get a new copy of the DNA here. This new copy of the DNA, actually what do this new copy of the DNA will this new copy of DNA will expel out this old DNA. So they will dissociate this old DNA. They will expel out this old DNA. Once they expelled out this old DNA, and no, this new DNA strand which are formed from this upper strand, they also contain the proteins, and we call that proteins 80 kd 80kd terminal proteins. Fine. Now what happened in this case? Now try to understand the concept. First of all, these two strands are present and the upper strand is used as a template, and from this upper strand a new DNA strand, means a new single strand of the DNA are formed, which can dissociate or which can replace this DNA. Once they will replace this DNA, so only the upper DNA will be around with this old DNA. Fine. So what we get just like that structure. From this we get this one structure, and also we get the other strand will also, the other strand will be dissociated. Fine. Now the other strand will be remain out from the nucleus. So once they will remain out from the nucleus, and what happen? So once they will remain out from the nucleus, this strand will having the self-annealing property. So this strand will self-annealing their self, and they will make a pen handle like structure. So we get a pen handle like structure just like that. These are the nucleotides. So we get just like that structure. Once we get this like their structure, pen handle like structure, now this strand is used as a template, and then we get this one strand again, these two strand again. These two strand we get again from this pen handle like structure. So now let me write pen handle structure, and we get this one genome once again. But actually these genomes are not required. Why? Because the actual genome of the adenoviruses are this one, 55kd terminal proteins. So this genome means the adenovirus have the genome, but with the genome they have 55kd terminal proteins. But in this case, one strand will carry the 80kd proteins and the other strand will carry 55kd protein, and we don't need this one strand. So what happened? After that what happened? This one protein sites or this one protein site will be cleaved by some enzymes, and once they will cleave by some enzymes, and they will replaced what? They will again replaced this old protein, means they will cleave the 80kd proteins and replace the 55kd terminal proteins again. So again we get the 55 terminal proteins, 55kd terminal proteins. So now this is the actual genome that we required. Now from this actual genomes, once these actual genomes are formed, then what we get? We get late mRNA. This blue one represents late mRNA, and from late mRNA what we get? We get the structural proteins. Now then we get the structural proteins. These are the structural proteins, and these are the late proteins, we also call that, or these are those proteins which are responsible to form a capsid, means which are responsible to form a pentose and hexone area of the capsid. Fine. So these are the capsid, these are the proteins which are responsible to make the capsid, means to make the hexone portion of the capsid and pentose portion of the capsid. Fine. So then we get the capsid area. And within capsid there are the hexones, are also present, and the pentones are also present. Fine, but the spikes are not present. Where they get the means the glycoproteins or the receptor proteins, or we can say the receptor proteins are not present, attachment proteins. Where they will get these attachment or the receptor proteins, after this capsid will form, inside the capsid there are the double-stranded genome are also. Once these capsid area are formed, then they will migrate to the Golgi body and endoplasmic reticulum, and from Golgi body and endoplasmic reticulum, then they will get what they will get, means after that we get a progeny adenovirus, or we get progeny virus. So now let me draw the structure once again. So we get just like that structure of the adenovirus. These are the spikes and these are the proteins, attachment proteins. Fine. So then we will get the mature virus, or we can say that progeny virus, and these are the hexos. This is the double-stranded DNA. Finally, we get the progeny virus. Now this mature virus will put pressure on the host cell, and by which the host cell will burst. These viruses will cause the destruction of the host cell. They will kill, because these viruses are naked viruses, and I told you the naked viruses will kill the host cell, and once they will burst the host cell, then a lot of the viruses, adenoviruses are released out, and they will ready to attack another new host cell and start their life cycle again and again. Fine. So that's how the adenoviruses will start their life cycle. Now explain me the whole life cycle with the in Urdu और इन हिंदी लैंग्वेज. ये आपके पास एडिनो वायरस का स्ट्रक्चर है, ये आपके पास नेकेड वायरस है. इसका जो साइज़ है वो आपके पास, नाउ द साइज ऑफ दिस वायरस आर 90 to 100 नैनोमीटर. ये इसका साइज़ है. सही. दीज आर द साइज ऑफ द एडिनो वायरसेस. क्या होता है, सबसे पहले ये जो होस्ट सेल होते हैं, मतलब एडिनॉइड टिश्यूज़ जो होते हैं, इसमें होस्ट सेल भी होते हैं. तो क्या होता है कि ये चूँकि एडिनॉइड टिश्यू से सबसे पहले हासिल किया गया था, तो इसलिए इसका नाम एडिनॉइड वायरसेस रखा गया, और यह असल में कैंसर कॉज करता है. सही. ये होस्ट सेल जो कि होस्ट सेल के ऊपर कार फैमिली रिसेप्टर होते हैं, ये वायरस इसका जो अपना रिसेप्टर होता है इसके साथ अटैच हो जाता है, और फिर ये अंदर पूरा का पूरा वायरस चले जाते हैं. सिर्फ ये ऊपर जो प्रोटीन एरिया होता है ये बाहर रह जाता है और वायरस अंदर हो जाता है. फिर ये वायरस जो है माइक्रोट्यूब्यूल्स में चला जाता है. माइक्रोट्यूब्यूल्स में ये न्यूक्लियस के जानिब पहुंच जाता है. वहाँ पर जब ये न्यूक्लियस में जाते हैं तो इसका जो कैप्सिड पोर्शन होता है वो बाहर रह जाता है और सिर्फ डीएनए अंदर आ जाते हैं. इसके डीएनए के टर्मिनल साइड पर आपके पास, या इस डीएनए के जो टर्मिनल साइड होते हैं वहाँ पर 55 kD प्रोटीन्स होते हैं. फाइन. नाउ फिर इसके बाद क्या होता है, ये जो डीएनए होता है यहाँ पर चूंकि न्यूक्लियस के अंदर आरएनए पॉलीमरेज एंजाइम होते हैं, तो फिर हमारे पास क्या बन जाता है, मैसेंजर आरएनए, अर्ली मैसेंजर आरएनए बन जाते हैं. अर्ली मैसेंजर आरएनए से हमारे पास प्रोटीन्स बन जाते हैं E1, E3, E4. ये प्रोटीन्स असल में E1 हमारे पास ट्रांसक्रिप्शन को कम करने के लिए इस्तेमाल होता है, E3 और E4 रेप्लिकेशन के प्रोसेस के लिए इस्तेमाल होता है. सही. तो फिर क्या होता है, ये स्ट्रैंड जो है, ये प्रोटीन जो है, फिर अगर मैं इसको यानी इस तरीके से फिर ड्रा करूँ तो क्या होता है कि ये स्ट्रैंड, ऊपर वाला स्ट्रैंड टेम्पलेट के तौर पर इस्तेमाल होता है, और इससे हमारे पास क्या प्रोड्यूस होता है, ये नया स्ट्रैंड प्रोड्यूस होता है. ये नया स्ट्रैंड जो है इस पर आपके पास 80 kD प्रोटीन्स होता है, और इस ऊपर वाले के पास आपके पास कौन सा प्रोटीन होता है, 55 kD प्रोटीन. तो फिर इसके बाद ये जो नीचे इसका अपना ये जो दूसरा स्ट्रैंड होता है ये क्या हो जाता है, निकल जाता है यहाँ से. जब ये निकल जाता है तो ये हमारे पास निकल जाता है, और फिर हम क्या हासिल कर लेते हैं, हम ये 2 स्ट्रैंड हासिल कर लेते हैं. अब ये जो है ख़ुद ब खुद सेल्फ इनीलिंग हो जाता है, मतलब ये ख़ुद ब खुद इस तरह अराउंड हो जाता है जिसके ज़रिए हम पेन हैंडल स्ट्रक्चर हासिल कर लेते हैं. अब ये पेन हैंडल स्ट्रक्चर जो है एज ए टेम्पलेट इस्तेमाल होता है, और ये पेन हैंडल स्ट्रक्चर जो है असल में ये क्या करता है, E1 और E3 और E4 प्रोटीन को इस्तेमाल करते हैं. E3 and E4 protein. They are actually use these E3 and E4 proteins. Now once they use these E3 and E4 proteins, फिर हम क्या हासिल कर लेते, हम इसी तरह स्ट्रक्चर, ये स्ट्रक्चर हम दोबारा हासिल कर लेते हैं। लेकिन चूंकि हमारे पास एक्चुअल जीनोम तो ये है, तो क्या होता है, ये जो पोर्शन होता है ये कट आउट हो जाते हैं 80 kd पोर्शन, और फाइनली हमारे पास यहाँ पर जो है 55 kd पोर्शन अटैच हो जाता है और हमें ये एक्चुअल जीनोम मिल जाता है एडिनो वायरस का। ये एडिनो वायरसेस का जो एक्चुअल जीनोम होता है फिर वो लेट मैसेंजर आरएनए प्रोड्यूस करते हैं। इस लेट मैसेंजर आरएनए से हमारे पास स्ट्रक्चरल प्रोटीन्स प्रोड्यूस हो जाते हैं। कौन से प्रोटीन? स्ट्रक्चरल प्रोटीन। अब ये स्ट्रक्चर प्रोटीन इसमें हेक्सोन और पेंटोस वगैरह होते हैं, वो कैप्सिड, तमाम तर कैप्सिड बन जाता है और फिर उसमें हमारे पास क्या हो जाता है, डबल स्ट्रैंड डीएनए। सही है। अब चूंकि इसके साथ ये रिसेप्टर्स नहीं होते, मतलब ये प्रोटीन्स नहीं होते, तो ये एंडोप्लाज्मिक रेटिकुलम और गोल्गी बॉडीज़ में चले जाते हैं और वहाँ से ये सरफेस रिसेप्टर हासिल कर लेते हैं, और या हम इसको कहते हैं ग्लाइकोप्रोटीन रिसेप्टर, ग्लाइकोप्रोटीन हासिल कर लेते हैं या स्पाइक्स हासिल कर लेते हैं। आखिर में हमारे पास क्या बन जाता है, एक कंप्लीट एडिनो वायरस बन जाता है, और फिर ये वायरस होस्ट सेल को बर्स्ट कर लेता है और ये वायरस होस्ट सेल को किल कर लेते हैं और ये वायरसेस निकल जाते हैं। अब ये वायरसेस दूसरे एडेनोइड या दूसरे होस्ट सेल को अटैक कर लेते हैं। तो ये था लाइफ साइकिल ऑफ adenoviruses. I hope you understand. If you have questions, so write it into a comment. I will try my best to give you answer. If you like the video, please hit the like button, share this video, and subscribe the channel to get more interesting video lecture like that. Thank you so much.